Thank you, Andrea. I will share my screen. Hello, everyone. First, I would like to thank Dr. Jerry Link and the organizers of the Open Box Science for inviting me to speak today. I'm very excited to be here and speak. I'm Noa Rachmian, and today I'll be presenting our study titled Identification of Senescent TREM2-expressing Microglia in Aging and Alzheimer's Disease. The work was done under the supervision of Professor Valery Krzyżanowski and Professor Michal Schwartz. And before we dive into the specifics, I want to acknowledge the efforts of my advisors and colleagues. I'm very thankful for all the help I received during this journey. So let's start. I assume most of you are familiar with the CNS, and you know that it's traditionally been categorized into two main groups of cells, the neurons and the non-neuronal cells, the glia cells. This group includes the oligodendrocytes, astrocytes, and microglia, which are the main focus of this article. Microglia are the innate immune cell of the brain. They're participating in removing dying cells and cell debris, provide brain protection and repair, and we are focused on them because of their crucial role in Alzheimer's disease pathology. Alzheimer's disease is one of the most common forms of dementia, affecting more than 35 million people worldwide, a number that is expected to get tripled by 2050. It's categorized by cognitive decline and memory loss, neurofibrillary tangles, amyloid beta plaques, synaptic and neuronal loss, and chronic neuroinflammation. Sadly, a cure for Alzheimer's disease is yet to be found, and age was found to be the major risk factor. One of the known phenomena during aging is the accumulation of senescent cells in tissues. Cellular senescence, which means to grow old in Latin, was first described by Leonard A. Flick and Paul Morhard in 1961 when they observed that ordinary cells in vitro would stop dividing after around 50 divisions. They called it the Hayflick limit. Cellular senescence is a steady state of cell cycle arrest that occurs in normally proliferating cells in response to various forms of stress, cellular stress. There are a few characteristics, as you can see in this slide. For example, you have the activation of cell cycle arrest machinery, including P21 and P16 upregulation, and the increased secretion of pro-inflammatory chemokines and cytokines, which called the senescence-associated synchronity phenotype, in short, SASP. The secretion of these pro-inflammatory chemokine and cytokine, the SASP, are assumed to be the reason for the senescent cells' negative effect when they are accumulating. There are a few triggers that induce cellular senescence, and in the short term, they have a beneficial role as tumor suppressors, and they are limiting tissue damage, and they have a role in embryonic development. Paradoxically, when they are accumulating, they're causing tumorigenesis and age-related diseases. It's the job of the immune system to eliminate them and prevent them from accumulating and have the detrimental roles. We also can directly eliminate them using senolytics, which are the drugs that aim to eliminate senescent cells. In this project, we used ABT737, which inhibits BCL2, BCL-XL, and BCL-W, which are anti-apoptotic agents that are upregulated in senescent cells. So when you inhibit them, you cause apoptosis in senescent cells and eliminate them, preventing them from accumulating and have the detrimental roles. So to wrap up everything I just told you to the hypothesis, we know that senescent cells accumulate during aging. We know that aging is a major risk factor for Alzheimer's disease. We know that senescent cells promote inflammation in tissues, and we know that there is chronic neuroinflammation in Alzheimer's disease. That led us to hypothesize that senescent microglia cells might have a role in Alzheimer's disease. And therefore, we first wanted to examine whether microglia exhibit senescent characteristic, and if so, if they have a role in Alzheimer's disease. In order to answer the first objective, we had to form a system that allows us to detect senescent cells. Detect senescent cells is not trivial because you need few of their characteristic for, being present at the same cell. Otherwise, it could be other different effects and not necessarily cellular senescence. That led us to decide to go to mass cytometry. Mass cytometry is, or CyTOF, it's a technology that allows you to use up to 40 antibodies at the panel in an experiment. Because instead of conjugating antibodies to fluorophores, they are conjugated to metals, and you have a variety of metals that can be detected based on their weights. So because we know that in aging in peripheral tissues there is accumulation of senescent cells, the first experiment and question was, do senescent microglia accumulate in old mice? So we took 24-month-old aged mice and compared them to young adult 4 months old. And we established a panel with 6 markers of senescent cells and 26 markers for myeloid and microglia cells. And we were really excited to see if we really did indeed detect senescent microglia, and if so, if they are accumulating in aged mice compared to young adults. So you can see here the heat map. You have on the left the different clusters that the t-SNE analysis gave, and on the bottom you can see the different markers that were included in the panel and the 6 senescent markers, different characteristics. And you can see in the t-SNE the different clusters, and encircled the senescent microglia green cluster. And in the bar graph, you can see the proportion of senescent microglia and how it's significantly higher in old mice compared to young adult mice. This is the proportion of senescent microglia out of all the microglia clusters, showing how indeed in aged mice there is senescent microglia and they are accumulating. But we wanted to check it in Alzheimer's disease, so we used the 5xFAD mice model, which has 5 humane familial Alzheimer's disease mutation, causing these mice to have amyloid beta plaques and neuronal and synaptic loss, cognitive decline, and neuroinflammation. We used the same panel we used for the aged mice, now in 5xFAD mice compared to age-matched controls. And we were able to, again, detect senescent microglia cluster that express all 6 markers of senescence. And you can see it in the t-SNE, in representative t-SNEs. And you can see in the bar graph how they accumulated also in the 5xFAD compared to the wild-type mice, showing that, yes, senescent microglia are accumulating also in 5xFAD mice. Visually, when looking at the heat maps, we saw that there is a big similarity across the different models. That led us to ask, are they the same microglia? Is it a conserved signature? So we analyze it using Pearson correlations, and you can see in the correlation matrix, cluster number 4 is the cluster of the senescent microglia, and it shows high correlation across the different models, meaning that indeed these are the same senescent microglia with a conserved signature across models. We also observed that they are highly expressed in TREM2, and that was really interesting to us because TREM2 is a very important player in Alzheimer's disease. It's a transmembranal receptor expressed by myeloid cells and microglia, and in neurodegeneration, it's helping the induction of disease-associated microglia, which are known to highly express it. Disease-associated microglia are present in context of diseases such as Alzheimer's disease and assumed to have a beneficial role. So when comparing the expression of TREM2 in disease-associated microglia to senescent microglia, we saw that senescent microglia are even upregulating their expression of TREM2 comparing to disease-associated microglia. And when looking at the correlation with senescent markers, we see that TREM2 expression is correlated with the different senescent marker expression. We also wanted to see if that means that TREM2 is affecting the accumulation of, if it has a role in the accumulation of senescent microglia. In order to test it, we knocked out TREM2 in 5xFAD mice AD mice model. When we compare the 5xFAD TREM2 intact and 5xFAD TREM2 knockout mice. When doing so, we saw that there was a significant reduction in the accumulation of senescent microglia following the knockout, meaning that TREM2 has a role in the accumulation of senescent microglia, but it doesn't explain the accumulation completely. And next, we further investigated the transcriptomic signature of senescent microglia by conducting a meta-analysis of previously published by Marco Colonna's group, single-nucleus RNA sequencing. The groups of the mice in this data includes wild-type, TREM2 knockout wild-types, TREM2 intact 5xFAD, and TREM2 knockout 5xFAD, just like the site of we did. We detected the cluster of senescent microglia in this data, cluster number 2, that appears in the TREM2 intact 5xFAD mice, but almost entirely absent in the other groups. This cluster was identified, as senescent based on the Sen-Mayo gene dataset and showed significant upregulation of TREM2, ApoE, CD9, and CD11C, similarly to the senescent microglia cluster identified in the on the protein level by the site of. Our second objective was to see if senescent microglia has a role in Alzheimer's disease pathology. So in order to do so, we used the Senolytic drug ABT737, which eliminates the senescent cells all over the body. We injected it 3 consecutive days IP and then tested for cognition and follow up by either CyTOF or real-time PCR. So let's see the CyTOF results. We can see that following the treatment with ABT737, there was a significant reduction in the senescent microglia accumulation. And when looking at the real-time PCR results, we can see that there was a significant reduction in the pro-inflammatory chemokines and cytokines. Many of them are familiar as the SAS of senescent cells. And when looking at the cognition, we use the novel object recognition task. In this task, the mice is subjected to 2 identical objects. We explore both of them. Then at the test, we replace one of the objects with a different one, a novel one, and we test the mice exploration of the novel object. Mice that remember they already explored the old one would now spend more percentage of their time exploring the novel object. And we measure it by the percentage of time exploring novel object. Mice that don't remember would spend 50% of their time with the novel and the old one. But if they remember, they would prefer it and spend more time with the novel. So you can see in pink that the wild-type mice preferred to explore the novel object, while the 5xFAD did not prefer any of the objects. They spent equal amount of time with both of them, exploring both of them, and following the treatment, the 5xFAD ABT group behaved as good as wild-type, preferring the novel object. So what it means, to sum up this figure, is that the senolytic therapy ameliorated Alzheimer's disease pathology in terms of cognitive decline and neuroinflammation. So to sum up, aged mice and AD mice accumulate senescent microglia cells with a conserved signature. Senescent cell marker correlated with the expression of TREM2, and I didn't show it to you, but in the article you can see that also of APOE. When knocking out TREM2, AD mouse models show reduced levels of senescent microglia, and senolytic therapy ameliorated Alzheimer's disease pathology. I want to again acknowledge the efforts of my advisors and colleagues and everyone who helped me during this PhD journey. Everyone in these slides helped me somehow during this time. And again, for you for listening and for inviting me to speak here today. I'll take questions now.